Molecular Biology Technique. I'm Sudha Naidu from BSc Biotechnology third year. It is a great pleasure and privilege to introduce our distinguished speaker for today's session, Dr. Sweta Sanguri, an accomplished life scientist and passionate science communicator who has been actively contributing to both research and public engagement in science. Dr. Sanguri is currently working as a scientific content creator at Science Nexus, where she plays a vital role in transforming complex scientific concepts into clear, engaging, and educational content. Through her work, she contributes to promoting scientific literacy, research awareness, and public understanding of modern biological sciences. Her efforts help bridge the important gap between laboratory research and society by making scientific knowledge accessible to students, educators, and the wider community. Dr. Sanguri holds a PhD in life sciences, during which she developed strong expertise in molecular biology, microbiology, and biotechnology. Her doctoral research involved advanced laboratory techniques and a deep exploration of biological systems, equipping her with a strong foundation in modern experimental biology and research methodology. Her academic journey reflects a deep curiosity about life sciences and a commitment to advancing scientific knowledge. In addition to her research experience, Dr. Sanguri has developed a strong interest in science communication and educational outreach, recognizing the importance of effectively communicating scientific discoveries to a broader audience. She has dedicated her professional efforts to creating high quality scientific content, educational resources, and knowledge dissemination platforms. Her work includes developing research based articles, scientific explanations, and learning materials that simplify complex biological concepts while maintaining scientific accuracy. Dr. Sanguri's work is particularly valuable in the present era, where effective science communication plays a crucial role in addressing misinformation, promoting evidence based understanding, and inspiring the next generation of researchers and innovators. By combining her scientific training with creative communication skills, she helps make science more engaging, understandable, and impactful. Through her professional journey, Dr. Sanguri exemplifies how scientists today can extend their influence beyond the laboratory by contributing to education, public engagement, and knowledge dissemination. Her work continues to inspire students and young researchers to appreciate the importance of communicating science responsibly and effectively. We are truly honored to have Dr. Sweta Sanguri with us today for this certificate course. Her experience in scientific research as well as science communication will undoubtedly provide valuable insights and inspiration to all the participants. Please join me in extending a very warm welcome to Dr. Sweta Sanguri. Over to you, ma'am.
Well, thank you so much, Sudha, for this kind introduction.
My pleasure, ma'am.
And it is always honoring to be here interacting with students, providing lectures. This is not my first time, not my second. I have been coming back again and again. And I'm extremely thankful that, you provide me with this platform. So I'll share my screen. And please let me know if you can see it. Is it visible?
Yes, ma'am, it is.
Okay, great. So I'll start. Very good afternoon to all the students and the faculty members present here. I would like to start with a simple question. My question is, if two patients have the same disease, should they always receive the same treatment? What would be our answer? Well, for most of the medical history, the answer was yes. But today, we know that even when the disease looks identical, under the microscope, their molecular signature can be completely different. And these tiny molecular differences can determine if one treatment will save a patient's life or it will do nothing at all to improve the problems of the patient. So today, instead of asking what disease that does this patient have, we are increasingly asking what is happening inside this patient's body, what is happening inside the patient's cells and the genes. Now imagine two patients walk into the hospital, both are diagnosed with lung cancer. And I'm sorry, I'm taking like in all through my lecture, you'll be having a lot of lung cancer examples, so bear with me. So these two patients are diagnosed with lung cancer. These both the tumors, they look very similar under the microscope. Do you think both patients should receive the same treatment? Well, for a lot of years, like for many years, this is exactly what the doctors did. There was same classical treatment which was given to patients of similar origin of cancer. But today we know something very important. Even though the diseases may look similar, the molecular changes inside each patient can be very, very different with each other. So these molecular differences, they determine how the disease will develop, how aggressive the disease will be, and whether one treatment will work on that disease or not. So this is the field of molecular diagnostics and precision medicine, which is transforming modern healthcare. So, for most human history, like I just said, medicine was practiced very differently from what we know today. Initially, a patient was visiting a doctor. Let's say 100 years back, the doctors had very few tools. There were no advanced laboratory tests, there were no imaging, and certainly there was no genetic analysis at that time. So, doctors, what they did, they would rely mainly on the symptoms. So, the patients will describe they have fever, they have cough, they have pain, and if they have fatigue. Now the physician, they will try to understand the diseases, and based on these visible signs, they will try to understand what kind of disease the patient is suffering from. So the treatment was largely guided by experience and the observation. Now later, medicine took a major step forward with the development of pathology. For the first time, doctors could examine the tissues under the microscope, and suddenly they could see some abnormal cells. They could find the abnormality, they could identify the abnormality, they could identify the tumors, they could detect the infections such as tuberculosis, and then they could better understand what disease looked like inside the body. But the scientists, they were still curious. They began asking deeper questions. So they began asking what is actually happening inside the cell. Why do these cells behave abnormally? So with the rise in molecular biology and genetics, we began to uncover these answers. So researchers, they discovered that many diseases, they begin with tiny changes in the DNA, and which in turn will cause changes in RNA and, of course, the protein, which will disrupt the normal cellular function. Now, this understanding has brought us to where we are today. We are now entering the era of precision medicine, where instead of treating disease in generalized way, we analyze the molecular profile of each patient, and then we tailor the treatment according to the molecular profile of the individual. So, if we look at the journey of the medicine, it has evolved step by step. First, we treated symptoms, then we studied tissues, and then we began to understand molecules. And today, we are moving towards truly individualized care. Now, what is molecular diagnostics? So, it refers to techniques that analyzes disease at molecular level. So it will include DNA, RNA, and protein. And by understanding and by examining these molecules, we can identify what is the underlying problem, what is the underlying biological changes which is causing the disease in the patients. So one important aspect of molecular diagnostics is that it helps in identifying specific mutations and biomarkers. What is a mutation? Mutation is changes in the DNA sequence. And what is a biomarker? Biomarker is a biological indicator that can be measured. We will talk about it in the coming slides. So detecting these molecular signatures, it can help us in identifying the characteristic of patient's condition. Another key advantage of molecular diagnosis is that it can enable very early diagnosis. Often molecular changes, they happen long before physical symptoms appear. And if we can diagnose these changes early, the doctors, they can intervene soon, and then they can significantly improve the patient outcomes. So these technologies, they are used in several important areas of medicine. For example, they are widely applied in cancer diagnostics, where identifying tumor mutation help to guide the targeted therapies. They are also crucial in diagnosing infectious diseases by detecting the genetic material of pathogens, like we have seen in the case of COVID-19 pandemic. RT-PCR was widely being used. In addition, molecular diagnosis, it also plays an important role in identifying genetic disorders which can be caused by inherited mutations. So overall, this molecular diagnostics, it allows us to look deeper into disease process, to move towards more precise and find it earlier, and also to design personalized healthcare. Now that we have discussed what, now comes the why. Why molecular diagnostics? Let me explain this with a very small clinical story. Again, lung cancer, like I gave a disclaimer in the beginning. So imagine two patients who come to the hospital, both are diagnosed with lung cancer. When the doctors they examine their tumor samples under the microscope, the cancer looked almost identical. Now, for many years, these patients would have received exactly same treatment, like I said earlier, because visually the disease appeared to be the same. But before deciding, in today's era, doctors perform molecular testing on the treatment. So when scientists analyze the tumor DNA, they may find something interesting. So in one patient, the tumor carries a mutation in a gene which is called as EGFR. And if you can recall this, if you can remember this, I promise it is going to help you in future, because these genes, cancer biology, cell division, this will keep on coming in your bachelor's, in your master's, and later if you go for PhD or for higher education. So if you all are a little attentive, and if you can remember these few genes that I'm talking about today, it is going to help you immensely in future. I can assure you that. So EGFR is, it stands for epidermal growth factor receptor. Now, EGFR, it is like an antenna on the surface of the cell. So, our cell, it is mostly independent, but to perform cell division, to divide, it is dependent on external signals. So, external signals are what? Different kind of growth factors. So, in surrounding of the cell, if the body needs more cell in certain area, there are signals which are sent to the cells, and these EGFR, they will receive the signal. These signals, once received, will tell the cell that now there is need for cell division, so you undergo cell division now. What happens when EGFR is mutated? It tells cells to grow and divide, and the signal becomes permanently switched on. It will now not need the growth factors externally. In normal body, in normal cells, normal lung cells, the signal is there. Whenever the signal will be available outside the cell, EGFR will catch the signal and cell will divide. But in case of EGFR mutation, no external signal is required. The receptor itself turns on permanently. So what happens when this EGFR is permanently switched on? You can think of it as a like a car where the accelerator is continuously pushed. So the cell is now receiving instruction to grow continuously. Now, here is where molecular diagnostic becomes extremely powerful. Scientists, they have developed drugs which are called as EGFR inhibitors, which specifically blocks this abnormal signal, which will block this EGFR abnormal signal, which is telling cell to constantly divide. Now, when the patient with EGFR mutation receive these drugs, the treatment can work remarkably well, because this is what is driving the cell division. But if the mutation is not present, the drug will not work because the mutation is not there. So without molecular testing, doctor will not know which patient will benefit from which treatment. This is the key idea behind molecular diagnostics. So I hope I'm able to explain why we are talking about molecular diagnostics and why we are going towards precision medicine. This is the base. This is what I wanted you all to know. We start from here, and then we will go further and try to understand more about molecular diagnostics. So now, instead of treating all the patients in same way and hoping for the best, we can now look at molecular drivers of the disease, and we can choose the treatments accordingly. So in other words, medicine is moving away from guessing and towards precision. So to understand molecular diagnostics, we need to revisit a basic concept in biology, central dogma of molecular biology. DNA is transcripted to RNA, and then RNA is translated to protein. So now DNA contains the genetic information. RNA carries that information, and protein, they perform most of the biological functions. So many diseases, they begin when mutation occur in DNA, which will lead to ultimately abnormally formed protein, which will disrupt the normal cell division or normal cell function. Now, by studying these molecular changes, scientists, they can identify the root cause of the disease. And when once the root cause is identified, targeting it or correcting it will become, of course it is challenging, but if we know the target, we can make a drug against it, we can make simplifying treatment, we can design therapies which will actually help to maintain the normal function where it has been disrupted. Biomarkers. Let's talk about biomarkers. So this word may sound a little technical, but the concept is actually very simple. Biomarker, it's just a measurable signal in the body which can tell us if there is anything wrong in the body or if the body is normal. So doctors, they use biomarkers in everyday in clinical practice. Sometimes they would not even call them that. A simplest example I could give is measurement of blood glucose level. So it helps to diagnose and monitor diabetes. So that is a biomarker. Similarly, when we check cholesterol levels in our body, it helps us to assess the risk of cardiovascular diseases. So a biomarker is a measurable biological indicator that helps us to detect disease. It can also predict outcome of the treatment, and it can also monitor how if the treatment is working or not. And there are several types of these biomarkers. Diagnostic biomarkers, they help us to identify if a disease is present. They will diagnose the disease. Prognostic biomarkers, they indicate how a disease is likely to progress. Predictive biomarkers, they help to determine which patients are most likely to benefit from a specific treatment. And pharmacodynamics, it shows whether a treatment is producing a biological effect, if the treatment is working actually in the way that we anticipated or not. So I'll give you an example here. For example, there is BRCA mutation, which is extremely common in the patients with breast cancer and ovarian cancer. So this can act as both a diagnostic marker and a predictive marker, which can help to guide the treatment decisions of a patient. Now, the question is, how do scientists detect these molecular changes? Several technologies make this possible. Some important ones I have included here: PCR, real-time PCR, microarrays, NGS. There is a lecture I took in NGS, I think 2 years back. If you are interested, all the students, you could find it. I'm not sure if it how it is saved in your university database, but if you look for it, I think you can find it. It is a very important session that has been covered by me last time. And fish. So each these technologies, they allow scientists to analyze genetic information in different ways. Some of these, they focus on specific genes, while other can analyze thousands of genes simultaneously. Let's go quickly for PCR. It stands for polymerase chain reaction, one of the most important techniques in molecular biology. So PCR, it allows scientists to amplify a very small amount of DNA. It can create millions of copies that can be detected and then also can be analyzed. So if you think of it, it can be like a molecular photocopy machine. So only a tiny amount of DNA is present. PCR can copy it and it can make it many times, copy it many times so we can study it. And a good real world example I already gave you is COVID-19 testing. During the pandemic, RT-PCR tests, they detected viral genetic material from the nasal swab samples, even when the virus was present in very small amounts. This is why PCR is considered highly sensitive and it is widely used in the diagnostics. Let's talk about next generation sequencing, or NGS. While PCR looks for one specific gene or one specific mutation, NGS allows us to analyze many genes at the same time. So you can think of it like, instead of reading one page of a book, NGS allows us to read an entire library, and it is simultaneously at the same time. So this technology, it can sequence millions of DNA fragments in parallel, and it can give us a comprehensive picture of genetic changes which is happening in one sample. It is widely used in cancer diagnostics to identify mutations that drive tumor growth, and It can also help us guide in targeted therapy, but its application, they go far beyond cancer. It is also used to diagnose rare genetic diseases where identifying a mutation can explain symptoms which were previously very difficult to diagnose. And it is also used in infectious disease detection to identify pathogens and also track disease outbreaks. It can play important role in prenatal testing, also in pharmacogenomics, and to understand the inherited diseases in some families. So NGS is a powerful tool of our time. It can help us understand disease at a genetic level, and it enable more accurate diagnosis and more personalized treatment. Now let's look at how molecular diagnostics is used in cancer. Cancer is fundamentally a genetic disease, meaning that it often arises due to mutation in certain genes, and these genes which control cell growth and survival of the cells. So, by identifying these genetic alteration in tumors, we can choose targeted therapies, like I explained for EGFR mutations. So if we can find these targets, we can design targeted therapies which will be much more effective for individual patients. So let's look at these few examples I have put here. So the first example here is EGFR mutation. It is commonly seen in non-small cell lung carcinoma. These mutations, these keep the cell signaling pathway continuously active, like I just explained. It tells cancer cells to keep dividing. So when we detect these mutations, a patient can be treated with the tyrosine kinase inhibitors such as gefitinib, which is specifically blocks the EGFR signaling pathway. So the patients who have this mutation can benefit immensely with this targeted therapy. Now, the second example I have here is BRCA1 and BRCA2 mutations, which is often associated with breast and ovarian cancers. So, the role of these genes is to help repair DNA damage. So, DNA damage is actually a very naturally happening process inside the cell. Our cells are continuously exposed to toxins in the environment. We are exposed to UV rays, we are exposed to lead, heavy metals, a lot of things which are bad for us, bad for our DNA. So when and also our lifestyle, sometimes stressful lifestyle can also lead to oxidative stress in the body, which can damage the DNA. So there are a lot of damage happening inside our body, and our body is equipped in such a way that our body is able to deal with most of these damages itself. So damage happens, body detects it, and then the cell, it there are many genes which help in repair of the damaged DNA. Repair happens and the cycle goes on. And somehow, if the cell is not able to repair these damage, the cells undergo apoptosis. So the damaged cells are removed from the system, and this is how normally how a body function or a living organism function. This is normal. So, in BRCA1, BRCA2, if the mutation happens, since these are the genes which help in repair of the DNA, if they get mutated, the repair mechanism will become defective. So they will not repair effectively. The cell will think that the genes are working, genes are there for the check, they will repair, but they are faulty now. And because they are faulty, these mutations build up and cause tumor. So, if these mutations are identified by the molecular diagnostics, these patients can benefit from PARP inhibitor therapy. So, these inhibitors specifically help the patients with BRCA1 and BRCA2 mutation, and mostly because these genes are these genes have a risk of running through the family. Hereditary risk for these cancers is very high, so this can be assessed. If someone in the family or few people in the family have ovarian cancers or breast cancer, they can get checked for BRCA mutation, and can be the tumor or the cancer or the risk of developing cancer can be identified very early on. Now, the third example is BRAF V600E mutation, which is commonly found in melanoma, skin cancers. So, when this mutation activates a signaling pathway which drives tumor growth, when this mutation is detected, the patient may benefit from BRAF or MAP kinase inhibitors, which specifically target the pathway. So the idea is to identify what pathway has the cancer cell chosen to grow. We can specifically target that pathway and we can block the growth of cancer. So this molecular profiling, it helps us to identify this specific mutation. And once we know, we can select therapy which will work, which will directly target that molecular change. And this is the foundation of precision oncology, treating the biology of cancer rather than just the location of tumor. Liquid biopsy. One of the most exciting developments in cancer diagnostic today is liquid biopsy. Please pay attention because it is going to be really interesting. So traditionally, if doctors they want to study a tumor, what do? They will have to perform a tissue biopsy, which means they'll have to surgically remove one small piece from the tumor. But scientists, they have now discovered something fascinating. So, what happens in a body when there is a tumor? It sheds tiny fragments of DNA into the bloodstream. And these fragments are called as circulating tumor DNA or ctDNA. Using advanced molecular technology like NGS, we can detect and we can analyze this DNA from a simple blood sample. So, all we have to do is draw blood like we do for other biochemical tests, and we would know if there is ctDNA present in the blood sample or not, and biopsy would not be needed unnecessarily. Noted. So, as a patient with lung cancer starts targeted therapy, and initially it responds very well. So the tumor shrinks, and the patient it improves, his condition improves. However, after some time, the cancer it begins to grow again. So initially the treatment was working, but after some time, the treatment stopped working. And now, why is it happening? So, in cancer, cells can evolve and they can develop new mutations in time that make them resistant to the ongoing treatment. It is a common phenomena with cancer cells. So now, instead of performing another invasive biopsy, doctors they can analyze circulating tumor DNA through the liquid biopsy. Now, this test may reveal a new mutation which is responsible for drug resistance, which will allow doctors to switch to a different targeted therapy or maybe a combination therapy with the old drug. So, just with a simple blood samples, clinicians can now track how the tumor is evolving and they can adjust the treatment accordingly. This is why liquid biopsy is considered a major advancement in modern precision oncology. Now, infectious diseases. Molecular diagnostics is also extremely important in infectious diseases. Now, traditional diagnostic methods they require growing microorganisms in culture. You must have also done that. Culturing microorganisms can take days sometimes. So molecular detection, they detect pathogens, genetic material directly, and which allows much faster diagnosis. And faster diagnosis means that doctor they will be able to start the treatment earlier, control the disease spread, and save a lot of time. And of course, the cost of the treatment can be also be saved. So now you have an idea about what precision medicine actually is. It refers to the tailoring medical treatment to an individual patient. So doctors, they will consider factors such as genetics, such as molecular biomarkers, environment, lifestyle of the patient. And the goal is very simple. Goal is right treatment for the right patient at the right time. Now, what's the difference between precision and technical traditional medicine? So, for the long time, medicine followed that what we call is one size fits all approach. So if the two patients, they were diagnosed with same disease, they would usually receive the exactly same treatment. In case of cancer, mostly the patients who are getting chemotherapeutic drugs. Now, chemotherapy is extremely toxic to the body. It kills the normal cells as well because it is not targeted. It will kill all the cells in the body. Of course, it kills the dividing cells with more specificity, or it is more aggressive against the dividing cells. But overall, the toxicity in the body remains. And there are extremely bad side effects of chemotherapy. We all have heard the patients, they get extremely sick, they get weak, they shed hair, and although it saves life, but if targeted therapy is available for some cancer or certain cancer type, it and if molecular profiling is done, and if one drug fits that cancer group, it would be advisable for the patient to take the targeted therapy because the treatment would be more effective, the toxicity would be less, it will also save a lot of cost to the patient. Now the doctors, they were diagnosing the same disease, and the patients were receiving exactly the same treatment, but something puzzling was happening. Not all the patients were responding to a treatment. Some with the disease, same disease, would show little improvement. Some will experience severe side effects and no, not much improvement. Why was it happening? So the answer, it lies in the biological and genetic differences between the individuals. Now each patient has a unique genetic makeup, and the diseases themselves, they can also vary at the molecular level. So this is where precision medicine comes in. Instead of treating everyone the same way, precision medicine uses molecular information such as genetic mutation, biomarkers, and gene expression patterns. And based on these, therapy can be tailored as per the requirement for each of the patients. So the if I have to say it differently, the goal is to deliver the right treatment to the patient. Like I have already mentioned, right treatment to the right patient and at the right time. Right time is what? Right time is as early as possible, as soon as possible, because this helps us in early diagnosis of disease that can save a lot of time and agony to the patient. So let me explain targeted therapy with simple clinical idea. In the past, cancer was mainly relied on chemotherapy. It would attack all the dividing cell. So while chemotherapy kills cells, it also kills normal cell. This is why patient experience side effects such as extreme weakness, hair loss, fatigue, nausea. So now we know there are different kind of mutations. Certainly, there are more that we still don't know about, but there are many that have been decoded with the advancement of molecular biology. So we know that many cancers, they are driven by specific molecular changes. Now, instead of attacking all the dividing cells, these modern drugs, they can target the exact molecule which is responsible for the tumor growth. So let's go for EGFR again. Lung cancer, mutation in EGFR gene. So it is continuously sending signals to the cell to grow. These EGFR inhibitors, they will block the signal and they will slow down the growth of the tumor. HER2 targeted therapy. Now, HER2 is again a similar receptor which is present in breast cancer. Sorry, the breast cells. So, in breast cancer cells, the tumor produces too many HER2 receptors on their surface. So, what happens, because this HER2 receptor is also going to catch the growth signal, and in HER2 positive breast cancer cases, the cell has made too many copies of these receptor outside the cells. And because the receptors are too many, they start sending constant growth signals to the breast tissue. So this HER2 targeted therapy, it specifically targets the cells which overexpress HER2 protein. So it will find these cells and kill the cells which are making more HER2 than necessary. So one of the targeted therapies is trastuzumab, which block these receptors, which and stop the signal, so the cell division stops. Now, there was a time when HER2 positive breast cancer was considered one of the most aggressive cancer type, very difficult to treat, and the mortality rate was very high. But by having these targeted therapies, the patient outcome has increased to many folds. Another example is melanoma. BRAF, we have already discussed briefly in the earlier slide. So for BRAF related mutations, which is usually in melanoma skin cancer, so these drugs are used, which are called BRAF inhibitors. They specifically block the abnormal signaling pathway like in HER2 and the EGFR pathway. So the idea behind the targeted therapy is precision. So instead of broadly attacking all the cells, all the rapidly dividing cells, as in case of chemotherapy, these drugs, they act like a precision strike. They will target the exact molecular driver of the disease. Pharmacogenomics. So now that we have identified the molecular pathways. We have known that this is the specific pathway which is causing the disease. We have the drugs. We can use the targeted drugs in the right dose, because having the right dose is also very important to get the maximum benefit from the treatment. So pharmacogenomics, it studies how genetic differences, it influences the drug response. So two patients which are receiving the same drug may not experience same outcomes. Some may benefit, others might develop side effects. So analyzing the genes which are involved in drug metabolism, doctors, they will be able to determine best drug for the patient, right dose for the patient, so that the treatment that the patient are going to have provides them with the best outcome. Precision medicine in oncology. So when we talk about precision medicine in oncology, the central idea is that cancer treatment is increasingly guided by the genetic makeup of the tumor. In the past, doctors mainly selected treatments based on where the cancer originated. For example, if the cancer was lung cancer or breast cancer or melanoma, there was same kind of drug that was being given to all the patients. Today, with the advances of molecular diagnostics, we can analyze the genetic mutations, we can profile the tumor, and this information, it helps us guide the treatment decisions. So I will once again talk about EGFR so that you all remember what EGFR is. Where does this kind of mutation happen? Mostly in lung cancer patient, non-small cell lung carcinoma. So if a lung cancer tumor, it carries EGFR mutations, doctors, they should prescribe EGFR inhibitors, which will specifically block that signaling pathway. Similarly, if breast cancer is HER2 positive, therapies like trastuzumab, which can target HER2 receptor, will be used. So by understanding these molecular drivers, doctors, they can select the drugs that can directly interfere the pathways that cancer cells will use, or they use for their growth and for the survival. And this approach is called molecular targeting because the therapy is designed to attack a very specific biological mechanism which is happening inside the tumor, which we have studied already through the molecular diagnostics. So, and precision oncology also uses several advanced strategy, which can help strategize the doctors to treat cancer more effectively. So these are targeted therapies which we just discussed, which blocks specific mutations. Immunotherapy, which we have not discussed here. These immunotherapy actually mostly help or strengthens patient's own immune system so that the immune system can recognize and it can destroy the cancer cells. And identifying which patient will get benefited from immunotherapy is also found out by the modern diagnostic techniques only, by sequencing, by NGS. So, precision radiation, where radiation is accurately delivered to tumor tissues surrounding healthy cells. So, when I say precision radiation, mostly hadron radiation is there, which can accurately be exposed in certain areas wherein it spares the surrounding tissues. So, when the right therapy is matched to the right patient, treatment will become more effective. There will be significant reduction in the unnecessary toxicity, unnecessary side effects. So, precision medicine, it is transforming oncology by shifting focus from one simply identifying where the cancer is located, because earlier it was like, oh, it is lung cancer. These drugs are given for lung cancer. This is ovarian cancer. These drugs will be given for ovarian cancer. But no, it's not happening like that now, because simply identifying the origination of cancer was not enough. Now, understanding what is driving the cancer is the at the molecular level is now being identified, and so the drugs are designed to fight that. Role of artificial intelligence. Now, this modern molecular diagnostics, it generates enormous amount of data. A simple NGS it generates data which we cannot actually, if we don't take use of certain softwares and AI, it is very difficult to derive meaningful information from the data that is being generated. Because if we are doing NGS, if we are simultaneously sequencing millions of genes, we will have millions of base pair of sequences. And to uh, identify where is the mutation of significance, it will take us forever if we don't take help. So modern molecular diagnostics, it is generating this data, and artificial intelligence, it helps analyze these complex data sets. So AI can assist us in biomarker discovery, the biomarkers which are not yet discovered, because it is evolving field. Cancer is the mutation rate is very high in cancer. So we cannot say that in certain kind of cancer only 10 different kind of mutations are happening. Maybe today there are 10. Maybe in future these cancer cells, they are extremely smart. They will stop responding to certain kind of treatment and they will get mutated in certain other way, and the previous treatment will stop responding. Then molecular diagnostic will again come into picture. A liquid biopsy will be done, or whatever biopsy is easy for the patients at that point of time, to identify what has changed inside the tumor, and a new treatment regimen will be designed. So AI, it can help in biomarker discovery, it can help in treatment prediction. if if already a treatment, if the results are pointing towards some kind of mutations, and then there is that there is sufficient information that that kind of treatment can help this category of patient or this kind of mutations, then AI can help in predicting what other treatment options are available. And AI now is also helping a big deal in drug development. So the time is really shortened, because a time for drug development is, it's a very huge timeline that is also being shortened with the role of artificial intelligence. Now, what are the challenges? Despite its promise, molecular diagnostics also faces challenges. These includes high cost And for a developing country like India, genomic sequencing technology, it can be really expensive. It can create significant barriers. There is also problems with insurance reimbursement, and not all our population is financially equipped to undergo genomic sequencing or to avail genomic sequencing. This is one. It is extremely costly at this point of time. Second challenge is data interpretation. So, like I said, the data that is generated is massive. It is huge data, and distinguishing between the significant data or mutations which cause problem, it can often lead to variants of uncertain significance, which we say VUS, and it is a barrier because VUS will not, it's a kind of in between. We know that there are variants which are different from the normal genotype, but we still don't know if they cause any pathogenicity, if these variants of uncertain significance, if they will cause any problems, if they are like that. So data interpretation is also very difficult because the data is huge. Ethical and privacy issues are there, a lot of concerns, because genetic privacy we might lose data security. And for employment region, for it can also have a psychological impact on patients if they find out something. Sometimes there are many benign findings, not just benign, some many incidental findings. Patient was living a very happy life, and suddenly a test came, and then the test showed that this patient might develop cancer, or maybe he's in early stages of cancer. It puts a lot of strain in an individual who is completely healthy and who's learning that he's diagnosed with cancer. There was no symptom or anything. It could have psychological impact. Not everyone wants to know what is going on with their life. And workforce shortage. So there is, of course, shortage of professionals which are trained in both the clinical medicines as well as genomics. We will generate a lot of data, but if there is no one to decode that data back, it won't make any sense. So even though if Reliance gives us a test in 1000 rupees and we all go and get the test done, so the question is, will we all get the significant results that we need? That is a question that we have to ask. The future of molecular medicine, it is extremely exciting. There are many emerging technologies, many ongoing technologies like CRISPR gene editing, which has revolutionized precise, targeted, and modification of DNA. It can sequence correct genet and correct it can correct the genetic defects. It can also treat inherited diseases at their source. So if we know that there is a disease a person has inherited, CRISPR can actually go in and edit that mutated version of the gene and make the patient healthy. Multiomics integration, where genomics, proteomics, and metabolomics, it is all the results from all these three studies. It is combined, and a view of, it's like a 360-degree view of a patient's health, wherein you can study all of it at once, what is happening, why it is happening. And the the person is not yet a patient. It's a person who has gone for some diagnosis, maybe a screening, and there is a 360-degree view which is available to the doctor. He can tell the patient, he can tell the person that maybe 5 years down the line, 10 years down the line, you might develop this kind of diseases. Or the doctor might tell him the nutraceutical profile of the patient, that if you take these kind of nutrition, it will help you in this way or in certain way. So a lot of decisions, lifestyle related decisions, can be taken so that in future the patient stays healthy, and it evades diseases which he might have gotten in future. AI driven diagnostics is again very exciting, and early disease protection that we have already discussed quite a few times during this lecture. Now, impact on healthcare. So traditionally, the medicine has always been reactive. Reactive in the sense the patient would develop symptoms, they will have some problem, then they will go to doctor, the disease will get diagnosed, and the treatment will begin after the illness has already progressed. So there was illness, we wait. It's how we do. So we get an illness, we wait for it to resolve for a few days, and then we go to the doctor. So the disease has already progressed. But with molecular diagnostics, we are moving towards a more proactive approach. So by detecting molecular changes in DNA, molecular changes in RNA or the protein, it is now possible to identify disease much earlier. So much earlier that the symptoms are maybe a few days apart or weeks apart or months apart. Sometimes the symptom might appear years later. But we know now that this is happening inside the body, and after some time, we will get sick. So it is exciting. So, and this is one of the advantage, early disease detection. And at the same time, precision medicine, it allows treatment to be tailored to individual patient based on the specific molecular characteristic, like in the lung cancer example. So when the right therapy is matched to the right patient, treatment will become more effective. There will be less toxicity. There will be lesser side effects, and the patient will recover faster. So ultimately, all of this, the goal of these advances is very simple: early detection, more personalized treatment, and better patient outcomes. So, as we come to the end of the discussion, one key idea stands out, which is molecular diagnostic. It forms the foundation of precision medicine. So by studying diseases at the level of genes, at the level of proteins and molecular pathway, we gain a much deeper understanding of what is actually driving the disease. This knowledge, it allows clinicians not only to diagnose condition, but they can diagnose it more accurately. They can also choose treatment which are better suited for the patients. And as these technologies they continue to advance, healthcare is gradually moving towards a model which is predictive, which is preventative, and which is personalized, where disease can be detected earlier, treatments can be tailored more precisely, and the patient outcomes they continue to improve. So ultimately, the goal is very simple: better science, which is leading to better care for the patients. Thank you for your attention. I can take any questions if you have.
I request participants to ask questions if you have any.
They can type in the chat box if they want. I would really like to see some questions here.
Thank you so much, ma'am, for joining.
Thank you for the invitation. I really enjoy being here with the students. I would have appreciated questions, but it is fine. They can send me questions later if they have. Please, you can circulate my email ID to them.
Yes, ma'am.